A bigger cursor. Hopefully that will help. And that was that I'm giving a seminar in Nebraska tomorrow, which is why I've got the fancy duds, I guess, today. And my stunt double, I don't see where he is now, will be giving a lecture on Friday. I will be posting this one normally. The other thing is, hopefully, those of you who noticed in D2L, I put quiz questions there. Those are exactly the same questions as we'll have in our clicker questions in about 2.5 minutes. Actually, maybe a little longer than that, because we'll talk about some of the questions from the exam. So if you want to take those, instead of being here, you can go and take them there on D2L. You have the time between now and the end of class to do that. You won't have the benefit of me discussing what the answers are, however, other than checking them out a little bit later on. I will be doing the same thing for Fridays so that Eric doesn't have to worry about all of the clickers and so on and so forth. So questions about that? Yeah?
Are we not doing in-class clickers on Friday?
There will not be in-class clickers on Friday, no.
Will Friday be recorded?
I am not going to be recording it, and Eric is not going to be recording it. Hopefully some of your colleagues will be, and I'm more than happy to post those to D2L if they can't do that discussion-wise. Yeah? How long do you have to have? Just during class, so none of you come. No. You'll have all day. But once you start those quiz questions, assuming I've set them up properly, there are 2 questions and you have 3 minutes. So it's just like what you have here. Other questions about this? It's kind of an experiment. I'm sorry I'm using you guys as guinea pigs for this, but that's part of the process. Any other questions, comments, worries? Yes, the material on Friday will be on the final as well. And I'll be asking Eric to give me some questions as well. I'll probably make up some as well, being based on that. But first, I did want to go over a couple of things from the midterm, because these are the questions that most people missed, and I wanted to discuss them in a little bit more detail. So the first one was A45, B32, C23, and you can back calculate, figure out how I manipulated the question numbers. A broken mRNA in bacteria leads to the production of a signal which is most similar to the signal produced by non-sense mediated decay, non-stop mediated decay, selenocysteine addition, program frame shifting, or polysome formation. And that's what this is. So tmRNA, also known as SSRA, although that's not important for this. But basically what happens here is what's down here in the corner. If we now go back, this is still the problem here. My big cursor still doesn't allow me to do all these things properly. Eventually we'll get this to work. What happens here is because you have this tRNA, which also has coding sequence next to it, you put this tag or signal at the end, which is basically the signal which says this protein is clearly an incomplete protein because it didn't have a stop codon. This mRNA is broken right here. And so what happens is that protein is tagged with this, and I always like to call it the eat me signal, to say this is clearly a bad protein, it needs to be degraded. In the case of the eukaryotic system, basically exactly the same thing happens here, only it's the lack of stop codon, but you still have a PolII tail. And so that PolII tail then is going to encode a protein that has all these lysines at the end, because that's what PolII codes for. And again, it's exactly the same way that you have a signal now for that protein, which says basically this needs to be degraded. Because this is clearly an incorrect protein because it didn't have the right stop codon up here. So that was the reasoning behind that question. The next one, that again, 80% of people miss these questions, which is why I think that it's worth going over them again. And I do think that they're important, otherwise they wouldn't have been on the exam. The protein that activates ubiquitin for addition to other proteins is called E1, E2, E3. The proteasome is ubiquitin kinase. As a professor, you love having these kinds of questions because the wrong answers are really easy to put in, E1, E2, E3. And then Nobody liked my ubiquitin kinase here. I think absolutely 0 people answered that particular question that way. But yeah, so this is all about this figure. And the ubiquitin activating enzyme up here is E1. Now, again, okay, fine. This is nitpicky, stamp collecting, insisting that you know all the tiny little details. But the important thing here is that this process of taking ubiquitin, adding it to this E1 protein through a high-energy bond is just like what's happening with your aminoacyl tRNA synthetases when they put amino acids onto the tRNA. It's not the actual chemical process is slightly different, but the overall goal is exactly the same. So it's this idea of activating an intermediate. And activation of intermediates, we've done it a little bit now in molecular biology. You get to cell biology, or those of you who are taking it now, activated intermediates are what it's all about. So that is sort of the reason that I came up with this question. There's also the ubiquitin conjugating enzyme. That's E2. These guys are the really general cases that you have in ubiquitin-mediated signaling. And then E3 is the one that gives you your specificity. So E3 is going to bind to the protein here that is going to be either degraded or just has ubiquitination, which has been added to it for whatever reason. Again, this could be signaling, could be all kinds of different things. It's not necessarily just the case for degradation. In this case, it is degradation, and it's the same proteins here which are bound to this as it gets a polyubiquitin chain. So the answer to the next question, which I didn't put up here, is how many different E3s do you need? It's always just one E3. And most people got that one, which I think was good. So the last one that I wanted to go over is this one. Assuming that we were able to select or design an RNA replicase that was RNA and could replicate itself, what's the next major missing piece in cellular life that probably arose about the same time as a self-replicating RNA replicase? And I was also thinking when I put these things together, maybe I should read all the questions for the exam. Unfortunately, we can't do that. But it's all about cellular life. And so the key to this question is really that cellular term here. And so self-replicating RNA replicases, these have to be separated from other self-replicating replicases, mostly having to do with metabolism, because if you have all your precursors, your mononucleotides, et cetera, that you need to put into your RNA for your replicase, but also for all of the other metabolic pathways, if you're sharing those with everybody else, it's not a very efficient system. And so you have to have some compartmentalization process. So that's the main thing here, and hopefully, again, this is the lead-in to the seminar on Friday in chemistry, 315-107 SB1. Lauren Roberts from Georgia Tech will be talking about the ribosome and how some of this fits into the, I think he calls it the origins of biology. I forget the actual name of the talk as well. So any more questions about the exam? There's one more thing that I wanted to mention is your score is not 50 minus the number you got wrong, it's the number you got right. And so people have had some questions about that. And the question that I X'd out because I made a mistake, that doesn't count as a plus point. Okay, questions, comments? I do have scantrons for people who haven't had a chance to pick them up. I'll have them in the back at the end. So now, the moment we've all been waiting for, or not, as the case may be, or you're leaving because you're taking them on D2L, which of the following is generally not regulated in gene expression? And let me actually turn this thing on. That might help. And we can start, I think, hopefully. There we go, yep. Replication, transcription, mRNA processing, protein modification, or protein degradation. And in case you are wondering, gene expression, I just noticed on here, is using what the book uses in terms of gene expression, their definition thereof.
So like that big flow chart? pretty much.
Yeah, that was what says gene expression.
That's what I said first, but they were saying that they don't think so. About processing and gene synthesis. I feel like that would be definitely a good, because it doesn't make sense to like go crazy. Okay, so
15, 10, 5, 'kay, we clearly have a bimodal distribution here. Replication versus protein degradation. Okay, clearly transcription, yeah, that's highly regulated. That's what we'll be talking about for the next couple of lectures, right, Eric? And messenger RNA processing, that's, taking your messenger RNA, how you're going to differentially splice it. We'll be talking about that next week. Protein modification, that's the fastest way to get a difference in terms of your output. Protein degradation is also a way to regulate, again, and the broader idea of gene expression is how much of that final activity do you have at the end. And that's why I sort of mentioned it should have been sort of the definition that our textbook uses. Some other people would say gene expression is just transcription. But that's where most of the regulation is probably going on. But there's clearly different areas as well. So replication is, in fact, the correct answer. Yay. And so now we can move on to our next clicker question, which is, which of the following is the fastest way to regulate gene expression? Replication, transcription, mRNA processing, protein modification, or protein degradation?
definitely not replication. It's either modification or transcription. I'm guessing. No, not necessarily. Lydia?
30.
So, to modify versus to degrade.
So degradation control as the one rate slower than
15,
So then it would be
so I'm
so modification, yeah.
10, 5, 4. So I wonder if I basically just told you the answer in my discussion to the answers to the last one, which I probably did. So when you rewind the tape, you can listen and hear that. But yes, it's the most rapid way to change the actual activity of any particular gene, or more specifically, it would be your gene product. Whatever's being made from that gene is going to be protein modification. Usually this is going to be phosphorylation, could be ubiquitination, could be binding to GTP, although that's not really a modification. But very much what most people think about here is they think about kinase activity. So it's going to be modification through phosphorylation. Okay, so let's move on and talk about regulation. Excuse me, if you're going to leave right after the clicker questions, you should really be taking them online. Okay? That was the point. So I guess it didn't work terribly well. There are a lot fewer people here today than there have been on other days, so hopefully that will help. So yeah, what are we going to talk about today? I wanted to start out talking a little bit about methods, which is kind of jumping ahead a little bit in terms of the way the textbook presents some of this data. So some of this stuff is from Chapter 8. If you want to go back and take a look at that, I'll try and remember to give you the actual page numbers of where those are. And then we're going to also change the order a little bit because I got poor Eric to prepare the bacterial transcriptional regulation stuff, and then I thought, oh, I could talk about it today so he can give you a brand new lecture that he hasn't prepared on Friday. So I decided that probably wasn't such a good idea. So the concept is clearly a very different one. It's going to be talking much more about the basic concepts of regulation, activation versus repression, and really what's best known in some of these bacterial systems. And so today we'll talk a lot more about the eukaryotic systems, particularly having to do with promoters, and then of course the main issue that we have with eukaryotic regulation has to do with chromatin. It's particularly the transcriptional level. Then we'll talk a little bit about insulators. One of the things that happens with eukaryotic regulation is there's a lot of regulation at a distance, and that actually goes back to when we watched that video where we had the assembly of the pre-initiation complex and then this thing that came from outer space, or at least from a distance, that sort of kicked off transcription taking place. And so there's a lot of action at a distance, but action at a distance can be a real problem if you think about what genes you want to activate and what genes you don't want to activate, because a lot of genes you want to not have be activated in different times, and particularly in different cell types, etc. And so this is a really critical aspect of thinking about how gene regulation works in eukaryotes, and we don't understand it really that well at this point. But basically it's a way of making sure one gene is regulated by one set of transcriptional regulators versus other ones. And then we'll talk about some of the regulation paradigms, but as I say, that's probably mostly going to be what Eric will talk about. Okay, start out with methods, assuming that this thing will work. First one is the electrophoretic gel mobility shift assay, also known as EMSA. Most people will call this a band shift assay. Basically, what you're looking at here is proteins bound to DNA. And what happens when you have proteins bound to DNA? That DNA gets bigger. So I should say that complex is bigger. Because that complex is bigger, it moves through a gel slower. And so it's basically what we're looking at here. Here's the cartoon version on this side. Should get my cursor to work here. No, why is it so small? It was big before. Over here. Is that better? Yeah. Macs, what can I say? Although I much prefer them to PCs. So here we have a DNA of a particular sequence. You've gotten it from somewhere. And you know how big this particular piece of DNA is. And if you put it on a gel, it will go to a certain position after a certain period of time. Then you incubate this piece of DNA, and it's a whole bunch of different pieces of DNA, with a mixture of a whole bunch of different proteins. And so this could be, for instance, all of your general transcription factors, or all of the proteins that you happen to find in a cell at a particular time. You put all of these proteins together with this DNA, and there are many, many copies of this DNA. One protein will bind here and make a slightly larger complex. You'll also have this big protein here that binds up here and makes a bigger complex. And again, you separate these guys on a gel, which just separates them based on size, and you'll have multiple different bands. This is going to correspond to this small protein right here binding, this one to that next larger, et cetera, et cetera. This looks great in cartoon form. What it really looks like is what's over here. This is a real example of that. Here we have our DNA down at the bottom, because this is where it was going to be migrating to in the absence of anything else. And that should be, actually there's not a control here, but there would be another lane that has a control that has nothing in it. And then you're looking at all of these other complexes which have formed on this particular piece of DNA. And the way this experiment was done is it's a separation, and we'll talk about column chromatography again towards the end of the class, but each of these separate fractions, so it's taking all the proteins that are in that particular sample, separating them from each other, and then seeing which of those bind to DNA. And what you can see here is that there's, in these fractions, you have something which binds to the DNA and makes it a lot bigger. You've got some things in this fraction which bind to DNA and make it a little bit bigger. Some things in these fraction over here which bind to the DNA and make it just a tiny little bit bigger. And these are those kinds of fractions people will talk about where those factors came from originally. You remember TF2A, TF2B, TF2E, TF2F, etc.? Those all came from this kind of experiment where you had here, this would be fraction A, this would be now complex A, complex B, complex D, etc., as you move across one of these column chromatographs. So it's the electrophoretic gel mobility shift assay. This is an electrophoretic gel. Here is your normal mobility. The shift is what gives you these different mobilities. So you use this to find and usually purify proteins that bind to your DNA, your favorite DNA. And you don't know what those proteins are. On the other hand, what you can do is do this now much more specifically, and that's what's shown here in DNA affinity chromatography, where you have a whole mixture of proteins up here that you now try and separate based on what DNA sequence they happen to bind to. So the first thing that you do here is you'll take your whole mixture of proteins and see which ones of them bind to DNA in general. Because it turns out that most of the proteins in the cell don't bind to DNA. If you think about all your metabolic proteins, et cetera, those don't bind to DNA very much. So you remove all of these proteins here that don't bind to DNA and just hang on to the ones which do bind to DNA in general. And very often this DNA binding is going to be something like a completely artificial DNA, PolII, or something like that. Or in some cases even just something like heparin, which is a polysaccharide that looks a lot like the backbone that you have in DNA. So it's a very non-specific process here. And you end up with a mixture of these DNA binding proteins, including the one that you want, which are these supposed to be represented by these red spots right here. So now you take this mixture and put it on a separate column, which now has the exact sequence that you're interested in bound to that column. In this case, it's 5' GGGCCC, and nice inverted repeat structure here, GGGCCC on the opposite strand. So you put this mixture of DNA binding proteins onto this column. All of the proteins are probably going to bind to it. And that was the point. You already selected them over here. All these are DNA binding proteins. But the ones which are binding specifically because of probably alpha helices in major grooves now that have the appropriate side chains to bind to GC base pairs in this particular case, those guys are going to bind much more tightly than these guys here. And so when you increase the salt conditions, you can do this in lots of different ways. You can change the pH. You can basically mess with that chemical interaction. In this particular case, you think about a high salt wash. Very often these interactions are going to be from your side chains. What kinds of side chains? Basic side chains, the ones that are positively charged, exactly. And so basically what you're doing here is you're competing that charge-charge interaction between the protein and the DNA with now a protein and the salt concentration. So now you end up with a protein which is bound or does bind to this very specific small sequence here. One of the things to point out here is that there are very few of these red dots in what you start with. And this is very true for almost all transcriptional regulators. They're the gene regulatory proteins. Very small amounts that are present inside the cell, but very large number of genes that are actually encoding them. And so pulling the needle out of the haystack is exactly what we're doing in this particular case here. So using the specificity of binding to get this purification process. So this is great if you have a DNA sequence that you know that a particular protein binds to, so like a Tata box, for instance. However, if you have a protein but you don't know what DNA it binds to, there are other ways to try and figure that out. And so this is one of those. It's called DNA footprinting. And basically what you're doing here is with a known piece of DNA that your protein binds somewhere to, and that binding somewhere, you know because you've just done your electrophoretic mobility shift assay, right? So you have this piece of DNA, you know that a protein binds to it, but you don't know exactly where it is. You have a protein that you think binds to a specific place. Now you want to find out where that specific place is. So the way that you do this experiment is you take your protein, you bind it to this piece of DNA, and then you chop this DNA into lots of tiny pieces. And that chopping process works great everywhere except where your protein is bound to the DNA, because that's going to block the chopping from going on in that particular location. So what you're going to end up with is a whole bunch of different DNAs of different lengths, but none where the protein is bound to it, because the chopping aspect is not going to happen there. So this gives you a footprint, because that's where that protein was. It's blocked the cleavage of the DNA from actually going on there. Now you have a whole collection of DNAs of different length, missing all of these here in the middle where your protein was bound to. Now you need to separate these, again, through electrophoretic mobility, usually done on one of these electrophoresis gels. You'll have all of these shorter pieces of DNA here, a bunch of longer pieces of DNA, and then this footprint in the middle. And this is an exact case of that down here at the bottom, where we've got now your DNA in the absence of protein, your DNA in the presence of protein. You can now go and sequence this DNA and figure out that your protein binds to this particular sequence right here. And this is, in fact, how people found out a lot of these things that we talked about last time. You've got half sites interacting with each other. Each of those binds to a particular protein. This is exactly how those things were figured out. So this is great if you have a relatively big piece of DNA. You want to find the exact position that your protein is bound to it. On the other hand, if you have no idea what this particular protein is binding to, there's another way to do this. And this process is actually very similar to the RNA selection process that we talked about before when we talked about the RNA world in concept. Basically, you have a huge collection of different DNAs. And when I say huge here, this is billions of sequences, sometimes even more than that. Now, with this whole collection of sequences, you put it together with your favorite protein that you're trying to find out what sequence it binds to. It's going to bind to one or relatively few of the sequences in these billions of different sequences here. You now separate this protein with the DNA bound to it. Almost always you'll go back and do this again, generate a whole more massive number of sequences that you will now check with your protein binding to. You do this a couple of times, and then you end up with a number of sequences here, a number of DNAs which are binding to your protein, some more than others. Then you sequence all of those. There are going to be a whole bunch of them. And then from that sequence, you can make a consensus sequence. You just add up all the positions, like some of the logos we've already talked about, we've looked at before in class, and that will give you what that particular protein binds to. And this is the process also called SELEX, because you're doing a selection for these particular DNAs that are binding here. Yeah?
So both of these are specifically for just determining what sequence that particular protein binds to?
Yes, exactly. So this is determining what sequence that particular protein binds to. It's also what specific sequence that protein binds to when you're just looking at that particular protein in the absence of all the other things which are around inside the cell. And that's what the next technique is about. Yeah?
How do you isolate just that particular protein?
So the question is, how do you isolate your favorite protein? I always call this, YFP, your favorite protein. Usually, this would be in a process like you've done your fractionation, your electrophoretic mobility shift assay. You've got that protein, you've purified it that way. Or what people do in the genomic age is you have this genome sequence. And in that genome sequence, you see, this looks like it's got a helix-turn-helix motif, or a basic helix together with a leucine zipper. I'll bet it binds to DNA. What DNA does it bind to? Okay, what I'll do is I will take this gene, express it in my favorite bacterium, usually, E. coli, and then you'll get lots and lots of that protein, and then you do an assay like this to try and figure out where that's actually binding to. Yeah?
So in the process of binding the protein to the DNA, running it through a gel to separate out that DNA, we're then somehow extracting that DNA from the gel to then create more copies of it?
So here, usually this process right here, so the question is, are you doing this through a gel and then getting the DNA out of the gel to redo this? Usually this is a process which we're doing not in a gel but in a liquid phase. And so you've got a whole mixture of DNAs here that you just put together with your protein, and then you separate all the DNAs that are connected to this protein. And we'll talk about how you do that in just a second. Yeah?
I had a question about the gel from the previous
Yeah.
slide. If you have protein bound to DNA and the gel is basically measuring the charge to mass ratio, what if your protein is charged?
Okay, so this actually, the question is, if you're measuring charge to mass ratio, what happens if your protein is actually charged in this particular case? So this is going back to this case, because in the footprinting, you actually have removed the protein from your DNA to start with, so that's not as much of an issue. But here, you have, you're separating. You're exactly right. It's not just the mass. It's the mass to the charge ratio, which you're separating in electrophoretic gels. So if you have something like phosphorylation of the protein, say this protein right here that's bound to this band, it actually is going to migrate in a different place. And people even use this to look at modifications of proteins bound to DNA. So you could have 3 different bands here, for instance, which would correspond to your DNA binding protein with 1 or 2 extra phosphorylations that's attached to it. So you would see that as well. Yeah, so it's not necessarily one band corresponds to one protein. It's one particular species. It could also be a whole complex of proteins. Think about TF2D, for instance. Massive numbers of proteins, all that are going to be in one particular fraction. Yeah?
Also on this slide, those segments of DNA, those are known? Those are sequence segments? Or are they just random?
Okay, so the question here is, are these known sequences of DNA or are they random sequences of DNA? You have to have one specific sequence that you're looking at here. You don't necessarily know what the sequence of that piece of DNA is, but a classic example would be a promoter. So you take your promoter sequence and you look at what things are interacting with that particular promoter sequence. Okay, here in this case, just to emphasize the whole questions about gels, one thing that I didn't emphasize here but probably should have is before you now do this separation, you have to remove the protein from this. So that you don't have these kinds of issues as you were talking about with any kinds of change or modification to that. Okay, so now we get to probably the best but hardest to do of these techniques. Because as I mentioned before, all the stuff we've talked about so far has been, getting back to your questions, we know what this DNA is that we put into our test tube with our known protein. Or it's a whole mixture of DNAs that we put together with a known protein. Well, what's really going on inside the cell? There's a whole bunch of different proteins. You got the whole genome, which is there. What's actually going on in terms of protein-DNA interactions? And so that gets us to chromatin immunoprecipitation, which is what's shown over here on the slide. Is there any way we can... should have turned the lights a little differently. Can you guys see this okay? I think it's more trouble for me at the front here. So now you have all of your proteins bound to DNA inside the cell at a particular time under a particular condition. So say you've used hormone to treat your cells with, or you're looking at brain of a developing mouse. So you take that particular point in time and take all the proteins that are bound to DNA and cross-link them to that particular DNA. So that DNA, all the DNA of your genome, all the proteins that are there, you treat them, you cross-link those two things together. Then you chop up that DNA in some way, make it into smaller pieces. And that could be either a DNase, so an enzyme which you use to chop it up. You can use chemicals to chop up your DNA. You can even use ionizing radiation, which we all know makes double-stranded breaks, to break up your DNA. And then isolate your favorite protein. So here's YFP again. And how do you get YFP out of this whole mixture of proteins and DNAs which are in here? The way you do this is immunoprecipitation, and so that's why the immuno's in here. You have an antibody to your favorite protein, or you have an antibody to a tag that you've put onto your favorite protein because it's your favorite protein. You've been working with it for years, etc. So with that antibody to your favorite protein, now you can separate your protein, which is now cross-linked to all the DNA sequences that it's interacting with in the cell at the particular time and place that you decided to do your cross-linking here. And now you can separate this. Usually it's a precipitation process. That's why it's called immunoprecipitation. Now you have just your protein bound to all of the sequences in the cell which it was bound to up at the beginning. You can remove the protein and now sequence this particular DNA that your protein was bound to. And then from that, compare it back to the genome sequence and say, hey, okay, now I know where this particular protein is bound. There's a question in the back. I'll get to you in a second. So the question is, why do this instead of looking at expressed RNAs? So the reason that you're going to do something like this is going to be you have a specific protein and you want to know what it's interacting with, as opposed to looking at expression of any particular RNAs. That's a different experiment. It's also a very interesting one to do. But here, we're just interested in what that one particular protein is doing. And a great example of this is there have been a lot of studies done recently where people look at where specific modified nucleosomes are bound to the DNA. And just look all the way through the genome, where are all these histone H3, K9, acetylated things bound to? And we may get to that today, we may get to it next week, where those things are. Yeah?
What is cross-linking and how is it different from binding?
So what is cross-linking and how is it different from binding? So this is a chemical interaction. And so you actually get bonds that form between your protein and the DNA. And the main reason for that is that very often, we already talked about molecular interactions with each other. There are lots of weak interactions, and they come and they go. If you're abusing this whole system by breaking open the cells, chopping up the DNA, you're going to be having their weak interactions coming off all the time. And so here you want to find, again, what was interacting at that particular place and time. So the cross-linking is a covalent bond which forms, usually you use formaldehyde. Yeah?
How do you get the antibodies for that specific protein?
So the question is, how do you get the antibodies for your favorite protein? Usually, you go back and use what we just talked about here. You've used some poor unsuspecting E. coli to make lots of your favorite protein, and you've taken that and you plugged it into bunnies and gotten their blood and got their antibodies back out. So that's one way of doing it. Now, the other way of doing it actually brings me to this modified protein here. And that is when you get the E. coli to make your favorite protein, you get it to make your favorite protein with a little tag on the end of it that you can buy antibodies to. And so this is a much easier way than plugging the poor little bunnies. So that is probably a much more common way that you do that. You have to be careful, however, with this little extra tag that you put on it doesn't mess up whatever your protein is normally doing. So you have to check and make sure that that's the case. But yeah, you can only do this if you do have antibodies to your favorite protein. The last thing I wanted to mention down here at the bottom is sort of the newer ways that people are using this. You can try and get the sequence from each of these little pieces that your protein is actually bound to. And that's what people are doing now, what they call ChIP-seq. So ChIP is chromatin immunoprecipitation, followed by sequencing. The other thing that you can do is you can do chromatin immunoprecipitation. And we talked about microarrays a little bit before, and that gets back to your question about expression of the individual messenger RNAs. These are all of the genes in your genome that you have little pieces of DNA that you put onto a little microscope slide. And if you want to come visit the lab, you can see our little microarrayers there. You can put 20,000 spots onto one microscope slide. And then you see where these sequences, instead of sequencing all of them, where they're going to bind to, and that'll give you an indication which genes are actually being involved in that, rather than doing the whole sequence analysis. This process, the chip-to-chip, there's a lot of this in the literature. Sequencing has gotten so cheap and so easy to do these days that most people have gone in this direction. But it's still, this is a very time-consuming and expensive process to do, not least of having your antibodies, but looking at all the sequences, getting enough of the DNA to be able to do this experiment with is much more challenging than something like the band shift experiment or the EMSA that I talked about before. Yeah?
So the capital IP is different from the lowercase IP?
So the question here is the down at the bottom, chip to chip or chip to seq? Yes, this IP here, capital IP, stands for immunoprecipitation, whereas the lowercase is microarray, so people also call these DNA chips, and so that's where that chip comes from. Thank you for pointing that out.
So why would you do this as opposed to the other two?
So the question is, why would you do this experiment as opposed to the other two? And I think you mean the SELEX and the mobility shift assay. So the mobility shift assay and the SELEX assay, and to some extent the DNA footprinting assay as well, all of these are things you do with just your favorite protein and just your favorite DNA, or your whole mixture of DNAs if you're talking about SELEX. That's all done in a test tube, which is great and wonderful, gives you a lot of information, but it may or may not correlate to what's going on inside the cell. So usually you'll do those experiments first, give yourself an idea what these things are interacting with, and then do this kind of experiment and say, okay, what's actually going on inside the cell with a whole mixture of proteins, all the proteins that are there, and your all the DNA in the genome. So it's a much better way of doing this, but it's a lot more complicated, difficult, expensive, etc. Okay, so finally we get back to transcriptional control. This is good, Eric. Now this is working perfectly for your lecture for Friday, right? So mentioned before, kind of keep beating on this, the major level of gene expression control is really here at the transcriptional level, again, going from DNA to RNA, because that's your first step. So if you regulate your gene expression up here, if you haven't turned on a gene here, transcribed it, you don't have to worry about any of the rest of this stuff. You're not going to have anything that's active. On the other hand, of course, it takes a while for anything that's transcribed to get all its processing and eventually make your proper protein. But the major level of control is at this level of transcription. So, talk about eukaryotic transcription. We have a number of problems with eukaryotic transcription. Most of them having to do with the fact that promoters are small and genomes are really big. If we think about eukaryotic genomes, our genome is a nice example of that. 3 billion base pairs in just one of our chromosome copies, 6 billion when you put them together. The other thing is these regulators, as I mentioned before, can bind a long way away from the promoter that they're interacting with. And we'll talk about these enhancers. We talked a little bit about enhancers already, but these are DNA sequences that these transcriptional regulator proteins bind to that are not right next to the promoter. RNA polymerase II, all of us remember from the last midterm, leads all of this alphabet soup transcription factors. TF2A, TF2B, TF2D, TF2E, TF2F, TF2H. And you need to put all of these together to make a pre-initiation complex. And each of these individual steps can be regulated. And then these eukaryotes have this annoying case of having all of this chromatin around. Why should we care? Well, a couple of reasons. I mentioned one of these right after lecture last time. The nanog protein that we talked about last time is one of 4 gene regulatory proteins that if you express in skin cells, you can make those skin cells become stem cells. And so basically, you can transform a skin cell into being able to make a whole organism just by the expression of 4 gene regulatory proteins, which is really kind of mind-blowing. Yeah?
Specific to skin cells.
No, these are not specific. These are not transcription factors that are specific to skin cells. These are transcription factors that you need in early development. So, yeah, and because of this, Susumu Tonegawa got the Nobel Prize 3 years ago now. Yeah?
Is that technically de-differentiation?
So the question is this technically de-differentiation? And you can think of that as one way of talking about it. Now, the differentiation process, there's a whole process that you go through. So it hasn't actually de-differentiated per se, but it's the expression of these 4 genes lead to that cell being in a de-differentiated state. But it's not a reversible process that you would have there.
Okay.
Yeah, so that's, again, it's really pretty amazing that just 4 of these can do this. And the experiments were published in 2009. They got the Nobel Prize 3 years later because this was pretty darn amazing. And other people were able to repeat it, which was very good. The other thing, and even more recently, I've been mentioning this ENCODE project a lot. This is basically, again, going through all the junk DNA, quote unquote, in our genomes and trying to figure out what's going on there. They came up with 636,336. No, I'm not going to ask you that number on the exam. But the binding sites in the genome for these gene regulatory proteins covering almost 8% of the genome, which is really amazing when you consider that the protein coding part of our genome is only about 1.5%. So all these binding sites are probably important for something. We don't know what they are doing in most of these cases, but they're clearly going to be important for something. And they're also, most of them are also pretty well conserved. So a lot of those are at promoters, and this ENCODE sequence found a little over 70,000 promoters. Wait a minute, but there are only about 20,000, 30,000 genes. What are all these extra promoters for? We don't know what all these extra promoters are for, but they're clearly there. So what do these promoters actually look like? We've talked about the Tata box, oops, back up, and where the general transcription factors bind, the BRE, the initiator element, the downstream promoter element. All of these are very close to each other. They're all right here within about 100 base pairs of each other. That's what also people will call the core promoter, where we've got all of our general transcription factors and where the RNA polymerase is binding. You will also have DNA sequences near where that is. And these are what are called cis regulatory sequences, again, within about a couple of hundred nucleotides away from where your transcriptional start is. But then you've got all of these other sequences that can be tens, and in some cases actually hundreds of thousands of nucleotides away from where your RNA polymerase is binding, giving you transcriptional initiation, which are still absolutely critical for getting transcription to take place at that particular promoter. You can even have these sequences, tens of thousands of nucleotides, downstream of where your transcriptional initiation takes place. And so these enhancers, is what they're called, where you have these regulatory sequences that you just put together, again, can be long distances away from the promoter, the core promoter, still activating. And so people call this the gene control region. So the gene control region, instead of being the hundreds of nucleotides or a couple of hundred nucleotides right around where the Tata box is, these are tens of thousands of nucleotides around where your transcription is actually starting. How do you get all these interactions? A lot of them are through our friend the mediator complex here, and lots of loops of DNA. And so that's partly where we had that loop that came in the video, where we have these transcriptional regulatory proteins interacting either through the mediator with everything which is here in your pre-initiation complex, or sometimes interacting directly. And this is just a reminder here again at the bottom here, mostly for me, that if you think about gene regulatory proteins, they're about 10% of the genome codes these regulatory proteins, but about 0.1% of the actual proteins are these gene regulatory proteins. So they're present in very small numbers. You remember the little red dots that we looked at when we talked about our purification process. Very few of those little red dots, but are having very major effects. So it's partly why you have to go through all these rigmaroles to figure out what your DNA binding proteins are, what they're binding to. Yeah?
How do you get the, those enhancers to associate with the mediator if they're so far away?
So the question is, how do you get the enhancers to associate with the mediator if they're so far away? So the enhancers are the sequences, and it's the enhancer binding proteins which are then going to interact with the mediator. And it seems to be kinds of things that we've talked about before. These are protein-protein interactions, lots of weak interactions, complementary surfaces that are interacting with each other. And those complementary surfaces may be modified proteins. They could have phosphorylation. They could have ubiquitination. Yeah, you name it, just to give you the appropriate interaction with usually a subcomponent of the mediator. Remember, the mediator complex is anywhere from 20 to 30 different proteins. And so each of those is going to have a specific interaction. And what you do see there as well, this is kind of getting back to here, is it's not just the mediator complex. Also, you have a lot of interactions with things like TF2D, and all of those, you remember the TAFs that were associated, so the TATA box associated factors that are part of TF2D. So TF2D is TATA binding protein plus TAFs. A lot of these TAFs interact with enhancer binding proteins. So it's helping bring that complex together. So another question here? Yeah.
So just a definition question.
Yeah.
So are enhancers and silencers not necessarily technically cis-regulatory elements then, because they're too far away to be cis?
So the question here is we haven't talked about silencers yet. You're getting a little bit ahead of ourselves. But certainly enhancers, are those still considered cis-regulatory elements, or are they trans-regulatory elements, just to paraphrase your question? Now, are they close enough to be cis, or are they far enough to be away to be in trans? Depends really on how you define cis and trans. I would call these in trans because they are so far away. And it turns out that one of the ways that people studied these enhancers and enhancer binding proteins associating with core promoter complexes is they actually put them on two different DNAs. Literally two different DNAs. So you have an enhancer on one DNA, and you have your core promoter on another DNA. And as long as they're linked to each other with catenanes, so we talk about catenanes, this is what happens when you have replication of a circular chromosome and don't have topoisomerase 2s. These DNAs are stuck to each other, even though they're not literally connected to each other in the same piece of DNA. If you have an enhancer on one piece, it can regulate a promoter on another piece. But if you separate these two, use a topoisomerase 2, this one can't do it anymore. So it clearly doesn't have to be on the same DNA in order to be able to regulate it. Again, it's pretty wild, and it immediately brings up the question of insulators that we're not going to get to today. But if you're going to act at a distance, why doesn't every enhancer binding protein interact with every promoter? And that's, again, a question we don't have a really good answer to yet. And what we do have answers to is this insulator process that we'll talk about in just a second. Another question here? Did I see some more hands? Okay, so this is just an overview, just looking in a slightly different way at what's going on here. You could consider the co-regulator here in the middle to be something like the mediator complex. This was what we would call proximal sites rather than cis sites. So you've got gene regulatory proteins right next to this pre-initiation complex, which is RNA polymerase II, general transcription factors. Here's our nice little TATA binding protein right here, binding to and bending the DNA. You also have these sequences a long way away that could be interacting directly with this pre-initiation complex or via something like this co-regulator complex, often the mediator, or you can also have downstream interactions here. All of that's involved in getting your polymerase to the right place. So what are these proteins that are binding to these, particularly these remote sequences? These are the gene activator proteins in most cases. You also have gene repressor proteins. But most of regulation, interestingly, in eukaryotes seems to happen through activation rather than repression. So these gene activator proteins often are going to be those that are binding to these enhancers. So again, they're enhancer binding proteins. A lot of people will use these terms interchangeably. Gene activator proteins bind to a specific DNA, and this sequence, again, it's an enhancer, so you can move it large distances away from your promoter and still have the enhancer binding protein there interacting with the pre-initiation complex, which is this, but it's diagrammed here as little red stripes. Here, well known for everyone's favorite eukaryotic system, which is yeast, exactly. The GAL4 protein, which is a yeast transcriptional activator, has two domains to it, and almost all eukaryotic regulatory proteins have two domains to them, if not more. But here it's got a activation domain and a DNA binding domain. And the domains, again, these are parts of the protein that fold up, have a particular structure, which then has a particular function. Normally, what's happening up here in A is that we have GAL4 binds to its regulatory sequence, activates your favorite gene over here. This is the normal situation. Now, what you can do by using your friendly E. coli or your recombinant DNA techniques that you learn in a lab next term down at the CLSB is you can now take your Gal4 protein and mix and match the domains. One of the great things about domains, they have a separate structure which will then have a separate function. And now what you can do is take your Gal4 and attach it to a DNA binding protein, in this case from E. coli. So you mix these two things together, make them, put them into the yeast cell. The yeast cell makes this protein, but it doesn't turn on this gene because the enhancer right here has the binding site for Gal4. Now, however, if you change this binding site to the binding site that this bacterial DNA binding protein binds to, now you get gene activation. And so what this tells you is that the activation domain, all you need to do is get it to bind to the DNA. And then it can play its role. So what do these activation domains look like? Well, we talked about DNA binding proteins last time. They're usually quite well behaved. They've got alpha helices, sometimes beta sheets. They're going to be interacting with the DNA, mostly in the major groove, sometimes in the minor groove. What are the structures of these activation domains look like? The answer is a mess. There's no nice structures of exactly how these interaction domains, they basically just seem to be sticky, which is, again, it's a really crummy term and not very good from a structural biology point of view. But it's this interaction that they have with the mediator or other parts of your pre-initiation complex. So turns out they are either rich in acidic residues, have lots of prolines, have lots of glutamines, but basically they're sticky. And so you bring a sticky sequence to the DNA, that interacts with something here at the promoter, could be the mediator, could be the TF2D complex, just trying to get your polymerase to this promoter. Once you've got the polymerase to the promoter, now you'll have transcription that starts. That transcription can very often be that first step in transcription that we talked about when we talked about transcription before, sort of an abortive initiation step, where it starts, it makes a couple of nucleotides, backs up, makes a couple of nucleotides, backs up. It's not had a conformational change where it's now in the elongating form, which is highly processive. So you have this polymerase, which is in the right place, and then very often we'll have the, oops, back up one more here, polymerase, which has been bound, so we'll see that here, so if I not advance a slide. Polymerase is bound. Here we've got everything bound to the promoter, but now this guy is just sitting here spitting out individual nucleotides. And so probably about 50% of human genes, and it depends on which numbers you look at, what cell type, what time, etc., that this complex now needs another transcriptional regulator to get this to move away from the promoter and move to that fully elongating state. And we'll see that this is the case when we talk more about eukaryotic regulation later on, that it's usually multiple gene regulators which you need to get full transcription to take place from one particular promoter. And I kind of cut you off before. You had a question?
It was related to the binding sequence and the
Yeah.
activator.
Mm-hmm.
Were you saying that via that experiment, they're kind of figuring out that they're separate, that you can like mix and match and you can activate different? Is that what you're saying?
Yeah. So the question is here, when I talked about that Gal4 Lex-A fusion, that you can mix and match DNA binding domains and activation domains. Yeah, that's exactly it. As you can have, if you can have, as long as you have a DNA binding sequence which is correct, whatever activation sequence you attach to it will activate at that particular place. Yeah.
So it's those particular sort of, well, I'll call it the top domain of that protein that's particular to a given gene being expressed as opposed to a particular location on the DNA that is causing that protein to bind that's regulating expression for that particular.
So I'll paraphrase your question and basically say the specificity for activation, is it coming from the DNA binding site or is it coming from the activation domain? Yeah. So turns out that it really is the specificity for DNA binding. It's not so much the specificity of the activation domain. And so if you have a DNA binding sequence that binds to a transcriptional activator, that will, if it's in the right place, cause transcriptional activation at one of those nearby promoters and nearby tens of thousands of nucleotides. Hey, you'll see Eric on Friday. You'll see me next Monday.
Are you doing office hours as usual?
Yes, office hours as usual today.